Hello, everyone, and welcome to the Botany and Plant Pathology Fall 2020 Seminar Series. Before we start, let me remind you of a couple of things. First, we will be recording today's session. Thank you, Dr. Gold, for allowing us to record this session. Also, if you have questions, please post them on the chat or raise your hand by selecting the Participants tab on and select the Raise Hand option. Today's presenter is Dr. Katie Gold, the newly appointed Assistant Professor of Grape Pathology at Cornell University, where she holds the primary research and extension responsibilities for grape disease management for New York State. She completed her PhD in plant pathology and master's in applied statistics concurrently at the University of Wisconsin-Madison in 2019. And her dissertation research pioneered the use of in situ and imaging spectroscopy for pre-symptomatic disease and diagnosis. Before starting her tenure track position at Cornell, she held a visiting faculty fellowship at the NASA Jet Propulsion Laboratory in Pasadena, California, with the carbon and ecosystem cycling and imaging spectroscopy groups to use hyperspectral imagery. She currently leads the Grape and Specialty Crop Sensing Pathology and Extension Laboratory at Cornell AgriTech. She also is currently combining plant pathology, machine learning, and remote sensing to study the fundamental and applied science of plant disease sensing to improve early disease detection and sustainable integrated management. Today, Dr. Gold will talk about her efforts to develop high spectral and spatial resolution measurements, data collection from autonomous ground robots to satellite networks and decision support systems. And without further ado, let's welcome Dr. Katie Gold. Dr. Gold, it's good to have you here. Thank you for joining us.
Thanks, Christian. Thank you. Thank you for the introduction, and thank you for the invitation. It's really great to speak to you all today. So as Christian said, without any further ado, let's get started on today's seminar. So I'm going to be talking about plant disease sensing, or the art and science, really, of studying host microbe interactions at scale. But oops, is this going to work? Yep, there it is. Okay, great. So first, why study host microbe interactions at scale in the first place? Why bother? So I first like to start with the statistic, which is that disease causes yield losses anywhere between 15 to 30% annually. And this equates to over $220 billion lost each and every year of the global harvest. So what could we do with that food if we had it? And what could we invest that money in if we had it? And in order to mitigate these losses, hold on, is this going to work? There we go. The United States alone applies well over 1.1 billion pounds of pesticides each year. So now our pesticides are absolutely critical for modern crop production and for ensuring the security of the global food supply. But their overuse does threaten biodiversity and is in direct conflict with the UN's recent edicts on sustaining biodiversity in the face of climate change. So in order to try to address these losses and develop a holistic solution, a global surveillance system for crop disease was recently proposed in a recent science paper. So this was published in 2019, and this outstanding group of individuals proposed a really wonderful organization, but I was really surprised when I found that they didn't even mention remote sensing for what and the role that remote sensing could potentially play in a global surveillance system for crop disease. So these are kind of the overarching things that motivate me and my program and what we study. And speaking of my program, that leads us to us and who we are. So I lead the Grape Sensing, Pathology, and Extension Lab at Cornell AgriTech, the Grape Spec Lab. And my program studies the fundamental and applied science of plant disease sensing to improve early disease detection and management. And as you can potentially guess from our name, we focus on grape systems. So plant disease sensing, as you can see by this cute little butterfly plot over here on the left, it's pretty complex, and it's just about as complex as plant disease is itself, which is a result of intricate and dynamic relationships between environment, the plant, the pathogen, as well as human management. That human management arm plays a really important part in how we sense disease, because we don't just let disease go in the field, we're actively trying to stop it. So some of the work that we do is about understanding how human actions are taken, or how the human actions that we take to stop disease impact our ability to find it in the first place. We receive our funding from diverse sources, ranging from NASA and JPL to the USDA, as well as the Cornell Institute for Digital Agriculture. And we're made up of a group of really wonderful individuals. We have members, affiliates, and partners all over the country. So I have to say it is quite challenging launching a research lab during a global pandemic. I started 5 weeks before the shutdowns began. But one of the benefits is that it's really taught us to be virtual and to be remote. And we run a program that spans the country and at this point, the world. So we have some wonderful members. We're anchored by my field specialist, Dave Combs, who keeps our vineyards alive. So we have something to sense. Nikita Gambier is a new postdoc with me. She comes from Nebraska, as well as my PhD student, Fernando Romero Galvan. His work is going to feature heavily in the first half of today's talk. And we have an undergraduate researcher, Graham Trolley, who's also contributing to the first half. And also he's on the market for a PhD. So if you guys like the work in the talk, I've got somebody for you. And we have a number of affiliates as well. So people who are primarily mentored in other programs, but are working with us on diverse goals. Josh Evans's tool is going to feature in the second half of this talk. And we have some wonderful partners as well. We have Ryan Pavlik at the Jet Propulsion Lab, who's our strategic university research partner. And he works directly with us in particular for the first half of the stuff we're going to talk about today. We work really closely with Yu Zhang as well, who's another assistant professor. He's our engineer at Cornell AgriTech, and our collaboration there is going to feature in the second half of our talk, and as well as partnering with Lance Keatle-Davidson and the Vitus Gen SCRI that he leads. And of course, there's me. So I really enjoy that my position allows me to kind of walk this line between NASA as well as in the field. So I spent every day this summer in the vineyard. Yet some days I'd be talking to growers and some days I'm talking to NASA program officers. So it's kind of cool, and I like that I get to walk that line. And I feel like this picture kind of sums up the line that I walk succinctly. So what are we going to talk about today? First, we're going to start with a little imaging spectroscopy 101. This is the type of remote sensing that we do. And then we'll move into our first case study, which is how do you sense something that you can't see, or how we sense asymptomatic disease detection. The next half of the talk is going to be on sensing something that you can't find, or our closed loop project that I mentioned briefly, we're working with Yu Zhang on, which is how we're trying to build this connected network between autonomous ground robots and satellite imagery. And then finally, if I don't dilly-dally too long on my slides, I'll give you a little bit of teasers of the 2 cool new projects being started by our postdocs. So now just to get all on the same page before I really get going, what is remote sensing? And I actually really like this definition that the USDA puts out because it captures it nicely. And that is that remote sensing is the process of detecting and monitoring physical characteristics of an area by measuring its reflected and emitted radiation at a distance. So basically, remote sensing is taking something in space or in the air, not on Earth, and using it to understand the Earth. And the type of remote sensing we do is called in situ and imaging spectroscopy. So on the top, we have the anatomy of light. All the way on the left, we've got our cosmic rays. All the way on the right, we've got our radio and our broadband range. And the range of light that we sense is this teeny tiny little slice in the middle. So every single color that we've ever seen comes from the visible light range. Now, this is the most abundant source of light from our sun, but it is only a pretty small portion. So we sense a ton of infrared wavelengths. And we also do a little bit of UV sensing. So now to understand our imaging spectroscopy products, which I'll go into just very briefly, there's really 3 different ways that we judge a product or we determine its usability. There are 3 different axes that we consider. The first of which is our spectral resolution, which is how many wavelengths does this device have? Or what is the range and what are the intervals at which it is measuring these wavelengths of light? I'm going to get back to that interval thought in just a second. The next is the spatial resolution. So how many pixels and how large are those pixels? The utility that you can get from a sensor that's delivering you 30 meter pixels is quite different from the utility that you can get from something that's delivering you 30 centimeter pixels. But the range that you get with those 30 meter pixels could be global versus your 30 centimeter pixels, maybe that's only a single plot. So that's another important axis to consider. And the final axis that we consider is temporal resolution, or how frequent the returns. So this could be something that you receive once a year at best, or something that you could receive almost daily, or maybe even diurnally. And something also important to consider as plant pathologists is that different pathosystems have different requirements for each one of these resolutions. It's not a one-size-fits-all when it comes to plant disease sensing, and I'm going to get more into that in just a moment. So I'm going to return back to that idea of intervals and spectral resolution. So imaging spectroscopy measures reflected light, but different imaging spectrometers measure light differently. So a multispectral sensor is our most common type of sensor. And this would be an NDVI sensor if you've ever worked with a red edge or something that's on a matrice. This is a multispectral sensor. Now, these guys measure light discretely, and they measure them generally in pretty large chunks. Now, this doesn't tell you anything about the range of light that they measure. You can have a multispectral sensor that's going all the ways out into the NIR, into that near infrared, but it measures light in discrete chunks. Now compare our hyperspectral, which is truly the imaging spectroscopy type that we utilize the most. These guys are a major improvement over multispectral in that they measure light at such narrow intervals that you can consider them a near continuous measurement. And the great power in these devices come when you get these continuous measurements over a wide spectral range. So we primarily work with sensors that measure light from the visible range into the shortwave infrared, or a range that's about 7 times larger than the range of light that we see with our eyes. So now how is this useful for plant microbe interaction sensing? So the overarching, I'm gonna cut right to the here. I'll unmute. Is it all good now? Someone nod their head. Yeah, okay, we're good. Okay, well now I'm actually gonna cut to the chase, which is that basically underlying disease physiology and biochemistry is what drives our ability to utilize these sensors in the first place. And the reason why they're so powerful is because they offer more information about plant microbe interactions than our eyes alone can say. So if we consider the leaf, light will enter the leaf and does one of 3 main things. It will be reflected, absorbed, or transmitted. Now this reflected light, these little blue arrows here in this diagram, they contain a ton of information about what's going on at the surface, but also the inside of the plant. So this light will go into the plant, leaves are designed to interact with light, bounce around and come back with all this really interesting spectral information about plant constitution. So now when we throw a pathogen into the mix, our pathogens physically and chemically change leaf constitution, and this will change the reflected light. But important to understand is that pathogens change plant constitution both in the immediate vicinity of infection, but also throughout the plant. So these little yellow dots here, these are representing effectors. So we know that pathogens release effectors that change the plant, and the plant is actively trying to stop pathogen interactions. And because of their signal-based immune system, this will result in somewhat systemic changes to the entire plant. So plant pathogen interactions both immediately change the vicinity of infection, but they also cause a systemic response throughout the plant. Now, when we utilize hyperspectral systems for them, we can rely on the knowledge that's been gained by the terrestrial ecologists over the years in the field of foliar functional ecology to actually try to parse out these spectral responses that we're seeing and try to identify the underlying constituents. So we know that different ranges of V-sphere light reflectance relate to different chemical and physiological constituents of plants, such as leaf water content. We've got nitrogen, lignin, cellulose, all of these things directly linked to reflected light. But also there's a number of things that link to reflected light that we can indirectly sense. So that might not have a direct relationship with a wavelength, such as let's say you've got nitrogen amount 20. If you have 20 nitrogen, well, you're going to have 20 nitrogen enzymes. Sorry, plant physiologists, I hope you don't hate me for saying that. So we can also leverage when we're utilizing these systems, those indirect relationships to inform our disease detection. But anyway, the one thing I want you guys to take away from this slide is that underlying disease physiology and these biochemical changes, as well as the physiological ones, these are the reasons why these sensors work. They're not magic, they're actually sensing real things that happen to the plant. And when we use imaging spectroscopy, we're allowed to scale this over wide regions. So we can scale this over spatial planes as small as the plane of an individual leaf, or as wide as the entire United States. And the true power of spectroscopy is that spectroscopy is really one of the only scalable ways we have to catch disease early. So we have outstanding active sampling tools for catching early disease. We have lip assays, PCR-based assays, ELISA strip tests that we can take out into the field, but all of these are reliant on us finding disease first. Spectroscopy offers a way for us to passively monitor for disease. Now, it's never going to be as accurate as a lab test, but it's a different tool and it operates differently and it has a powerful role to play. So, and we also know that these things are scalable. So we know that the fundamental aspects of plant disease sensing are scalable, but we also know that host microbe interaction itself is scalable with imaging spectroscopy. We know that beneficial interactions are scalable, such as sensing mycorrhizal association. So this is a figure that, if you'll note Romero Galvan, that's my graduate student. I'm very proud of him for his role in this work. These are from some colleagues at JPL who recently submitted a paper to PNAS showing that sensing mycorrhizal interactions in trees across different biomes is scalable with imaging spectroscopy. Additionally, we know that antagonistic plant microbe interactions are scalable as well. This is a figure from one of the most important papers published in plant disease sensing. This is from Zarka Tejada, 2018 in Nature Plants. This was the first paper to show that pre-symptomatic disease detection is possible at scale. So we know that both the beneficial and the antagonistic interactions are scalable, and that they're scalable because they're rooted in actual changes to underlying physiology that we can directly and indirectly sense. So just one last introduction slide before I really get into the meat of the talk. Just a little bit about our aerial and satellite spectroscopy data. So today I'm focusing on our remote sensing work. We have an entire arm of our program that's dedicated to ground sensing as well. But today we're talking about what we do in the sky. So we work quite a bit with AVRIS data. So this is the Jet Propulsion Lab's aerial and visible and infrared imaging spectrometer. And this is probably the catalog, probably the best imaging spectrometer ever made. It lives in California and the guys at GPL fly it if you pay them for it. So it's really great. It has the best spectral resolution and outstanding spatial resolution, but it has very poor temporal resolution. You might be lucky if you get a single flight. This makes AVRIS ideal for long latent phase diseases where you have a wide opportunity to catch asymptomatic disease, because that's our goal, to study and detect asymptomatic disease. It's also ideal for zero tolerance management threshold, and as well as sensors like it. So this is when you need to absolutely know if this disease is present or not, because there is absolutely no tolerance for it in the field. That's what makes this system so powerful. We use this one mostly for our California work. Now in New York, we work quite a bit with Planet data. So Planet Labs is a commercial satellite provider. They offer constellation networks. So this is a little artist rendering here. We work with their SkySat satellites, and these provide us with 50 centimeters from space resolution of almost anywhere we want on Earth daily. But you know, we do have to pay for it. Now these guys have really high temporal and spatial resolution. 50 centimeters from space is pretty good. But the caveat is that they have low spectral resolution. So they're nowhere near as good as the Cadillac AVRIS, but we get to cover a really wide spatial range at a really good pixel size, and we can have that temporal monitoring. So this makes it really good for short latent phase diseases, diseases that change very frequently that you need to be monitoring over time, such as oomycetes in my background that have explosive growth potential. And it also makes them ideal for our non-zero tolerance management threshold diseases, diseases where you do have that wiggle room to potentially allow for some misdirect. So now if you're working with potato late blight, you cannot let that sit in the field. But there are other oomycetes where it is okay to have a little bit. So you don't want to rely on this sort of data if there is a zero management threshold tolerance. I really want to emphasize that. Okay, so now we can finally get to our first case study, which is how do we actually sense asymptomatic disease? Oh, actually, no, I lied. I'm sorry. So we do have one more introduction slide, just one. So our asymptomatic disease work at scale was preceded by some really intensive in situ spectroscopy work that I did as a PhD student at the University of Wisconsin-Madison with my two outstanding PhD advisors, Amanda Gevins, who's a vegetable pathologist, and Phil Townsend, who is a remote sensing scientist and a terrestrial ecologist. So during my PhD, I found that pre-symptomatic disease detection is possible with in situ spectroscopy, and I worked primarily with potato late blight, which we see pictured here in the background. We also found that spectral biology, so that's a phrase that we use to capture how light reflectance changes according to plant constitution. We found that spectral biology actually captures different pathogen lifestyles, which forms the foundation of our ability to use these systems for pre and post-symptomatic disease differentiation. We additionally found that cultivar has a really strong impact on spectral biology and also pre-symptomatic detection accuracy. We couldn't just build a model in one cultivar and assume it would work everywhere. We found that different cultivars had different early spectral reactions to disease. We also, when we dug like super deep into the basic science of what could spectral systems do, we found that pathogen strain differentiation was possible, but this was only possible pre-symptomatically, which I thought was really interesting. And that actually spectral profiles and spectral biology converged as disease became more and more manifest and they became more and more diseased. So this was really interesting. But if we're working at the pre-symptomatic level, if we have a fine enough resolution data, we can do pre-symptomatic strain differentiation. And my final dissertation chapter, which I'm sitting on because it's hard to write during the global pandemic, but I promise it'll be out soon, is that we found that healthy upper canopy foliage on minimally diseased plants can be reliably differentiated from comparative foliage on non-diseased plants. So to summarize that, basically we found that if a plant has even a single late blight lesion in the lower canopy, we could differentiate its upper canopy foliage from a plant with no late blight lesions reliably with about 76% accuracy. And we hypothesize that this is linked to potentially activation of the plant's natural defense system or its attempt to try to fight off infection. So I'm working on this one. I'm trying to get it out the door. My two advisors are continuing this work, even though I left them. They have been awarded an NSF Biology Institute Award, the Spectral Biology Institute, and I'm really excited to see the work that comes out of this next. So if you're interested in potato late blight, do stay tuned with what comes out of the Gevins and Townsend's labs in the next couple years. Okay, so now we can actually talk about the disease that I'm doing in my own tenure track lab now. So one thing I want to start with first, I kind of seeded this earlier, is that our Avarice NG data, which is what I'm going to be talking about now, is ideal for our long latent phase diseases. So the work I'll be presenting here is my PhD student, Fernando Romero Galvan. Well, I guess the transition I really want to make is that I had this opportunity as a PhD student to do all this outstanding, really exciting in situ spectroscopy work. But what I wanted to do next was to learn how it would scale. So Cornell generously sponsored a short postdoc for me at the Jet Propulsion Lab, and I left JPL both with amazing ideas and great collaborators, but as well as my first student here, Fernando. So this work that I'm going to present on now, this is born out of that short postdoc, and I'm really excited about it. So we've chosen these long latent phase diseases because they're ideal for using our Cadillac sensor with, where we have maybe one or two opportunistic acquisitions over any given field. And the diseases that we identified as ideal candidates are first ESCA. Now this is a fungal disease, and this is actually a really nice model pathosystem for plant disease sensing because it's acquired in the nursery phase. So it allows us to mine historic acquisitions. We also know that plants with ESCA contain the pathogen within them for their entire lives after they acquire it, but they only display these characteristic visible symptoms about once a season, and they don't always display these symptoms. So it makes it a really ideal pathosystem to study plant disease sensing with. Our next two diseases have great industry importance in California. The first is grapevine red blotch virus, GRBV, and the next is grapevine leaf roll virus, which is caused by a complex of viruses. So I'll refer to these two as just the viruses. So I'm no virologist, I'm a plant disease sensing scientist, but these have outstanding industry importance and they have really long latent periods. So that makes them a really great target for plant disease sensing. So in particular, these two viruses together, they've reduced the lifespan of the average vineyard in California from 30 years down to 7 years alone, because there is a zero management threshold for these. And by the time you see these visible symptoms, it's already too late to stop the spread. You're really chasing falling dominoes. Because of that long latent period, by the time the first round of symptoms shows up, those plants were already infectious and spreading the disease to the rest of your field. So I'm sure all of us after the coronavirus pandemic can understand the danger that a virus with a long infectious latent period poses. Overall, these two diseases really pose, I would say, an existential threat to the existence of the industry, just by how quickly they're forcing us to turn over. It's not economically or ecologically sustainable. So this year we had the opportunity to make an emergency request, and we received a direct allocation from Woody Turner, the NASA Biodiversity Program Officer, who's really the hero of the hour, to support acquisitions of Avris NG over different vineyards in California. So we both have our data mining work, but we also have this new exciting data. So this campaign would not have been possible without our outstanding industry partners, as well as our partners in the UC system. So this campaign came together over the course of a weekend. I mentioned it was emergency because it was emergency. The fires were raging, and we saw an opportunity to get data that could potentially help both the viticulture and our own research efforts, but as well as the viticulture industry for years to come. Really sadly, so some of the vineyards that we flew in Napa as well as Sonoma, they were burned in the glass fire. So we hope that this imagery too will help in the future and encourage people in the remote sensing community to study problems that are linked to viticulture and agriculture more broadly. So we had industry partners over the course of a weekend commit to submitting over 30,000 data points. So actually, I think I did the calculation, 3 million individual vines of validation were submitted for this project. So I really have to thank our industry partners for it, as well as our Davis partners who went out into the field, our JPL partners who flew in the aircraft, especially because I was sitting in the Catskills. So this was Labor Day weekend and I was banned from travel because of Cornell. So I coordinated this entire campaign from the porch of our mountain house, this little vacation house, Airbnb we had rented. So without our outstanding partners, this never would have come together. So we're really excited about this data and we're just starting to parse through it. And here's an example here just of some of the data that we received from the study. So I want to highlight how teeny tiny these pixels are. So this is something that you don't often get with Avris. So Avris is generally flown at the 3 to 10, Avris NG is thrown around the 3 to 5, meter range, but they flew the airplane super low for us. So these are sub-meter pixel sizes. And here's this little inset up in the upper right. This is an example of the pixel size that you would normally get. So this is just a random band, and it's just showing the variation that we can see with these tiny pixels. And these dots here are where our partners took ground truthing information. I was really fortunate to be able to partner with Beth Forrestal at UC Davis, not UCSC. And she took foliar samples for multi-omic analyses too, to see if we could potentially link that to some spectral variation within those pixels, as well as Rachel Meyer, who is from UCSC, who collected eDNA and holobiome sampling. So we collected this across symptomatic and asymptomatic vines in order to see if there's a potential difference. So one thing I didn't mention is that these viruses, they're vectored. So, and we know in particular for leaf roll that leaf roll is vectored by mealy bugs, but the mealy bugs themselves are vectored by ants. So we suspect and we hypothesize that we could potentially develop holobiome markers that can help direct viral sampling and scouting. So this gets back to that difference between the active sampling and the passive monitoring. So we hypothesize that Avris can help us with better directing where we send our active sampling efforts, and that we can develop better active sampling methods that don't break the bank for our poor grape growers who are currently paying anywhere between $50 to $300 to get a single vine tested. So we're really excited to parse through this data. And then again, this map was made by my student, Fernando Romero Galvan, whose PhD work this data will encompass. So now this is that same plot there. This is our Lodi bank. It's kind of like the go-to one. We have really extensive ground truthing within here. So this is an example of our planet data. So this is what we work with in New York, but this is at 3 meter resolution. And this is NDVI. Oh, actually, I'm sorry. This is MSAVI too. So this is like MDVI, but it's better at adjusting for the really reflective soil that we have in California. So you can see here, there are some differences in MSAVI profile here. But if you look at that little inset to the left, you can see that the main differences identified by this index are just the gaps, and that we don't actually see that much variation elsewhere within the map. And this just goes to show why with a zero management threshold disease, why it's so important for us to have better spectral and better spatial information. So now this is our very preliminary, absolutely super preliminary, Fernando made it this morning, graph. So we're testing out some really cool unsupervised machine learning methods. This one here is unsupervised anomaly detection. So this is a kernel detection method that automatically scans. If anyone's played Among Us, it's basically like scanning all the crew members and automatically identifying the imposter. But this method relies on the assumption that the anomaly is not common within the set. But this set, we estimate that the virus incidence is around 2%, which is why we do believe it's ideal. Now, this pixel we haven't yet filtered out for those gaps that are already existing. So we can see that some of the large anomalies that we've identified are actually just where we don't have vines anymore, but we do see some smaller ones. So if we go back here, hold on. So if we go back over here, we can see this region around here. We can see that we have a couple of little baby hotspots up in there. So we're excited to keep working on these methods and dive into them, both to establish new utility for this method, as well as establish whether or not we could utilize hyperspectral reflectance at scale for disease detection moving forward. So I also want to highlight some of the cool work that our undergraduate Graham Trolley is doing. So Graham is actually working to build a geospatial database of historic adverse acquisitions over vineyards using the Cropscape, the cropland data layer. In order to further our own asymptomatic disease detection work, our ground and industry partners have committed to submitting validation data for anything that we may find we have data for, but as well as to facilitate more viticulture research with this powerful tool. So JPL has been flying AVRIS over California for years now, and it's been just happens to catch vineyard acquisition. So we hope that by building this database that we can also make this tool more usable for the agricultural community. And additionally, Graham's senior thesis, one of the things that we found in our work on the ground was that cultivar has a huge impact on early disease detection. So he's going to be comparing and contrasting different supervised and unsupervised methods for automatic variety or cultivar detection within this data set. So I'm excited for his senior research, and he's looking for a PhD if anyone's interested in him. So some of the next steps from this first case study. We're continuing our work with unsupervised anomaly detection, as well as comparing other both supervised and unsupervised machine and deep learning methods. We're also going to be doing a dimensionality assessment. So we want to understand how much variation indicative of asymptomatic infection captured in this imagery could potentially be captured in other types of imagery. As I've mentioned, this is really the Cadillac. AVRIS is probably the best imaging spectrometer ever built, and we've flown it lower than they've ever flown it before. We really can't get any better than this, but we are unlikely going to be able to have this imagery moving forward. What we will have is surface biology and geology. Now, this is a hyperspectral V-square imaging spectrometer that's going to be launched by JPL in somewhere between 5 to 6 years from now. And this will yield 30-meter imagery everywhere on Earth once every 2 weeks. So we want to best understand what we can learn from this really Cadillac AVRIS imagery on the ground to understand how much and how we will be able to utilize this forthcoming satellite the best. Oh, I keep getting, okay, we're good. Also, we're working with colleagues at Davis and JPL to build a triaging algorithm, and particularly we're working with the EcoStress team. So EcoStress is a satellite on the International Space Station. It provides thermal imagery at 70-meter resolution, but they've developed a method to downscale it with planet imagery to about 3 to 5 meters, which is really cool. So these vineyards are not just diseased, you know. This year, I'm sure you guys heard about the devastating wildfires in California. These vineyards are heat wave stressed. They are smoke stressed. They are just unhappy and thirsty. And we really need an ability to triage these different types of stresses in order to put them into the best pipeline moving forward if we want to best utilize these satellite systems for disease detection, as well as other applied agricultural monitoring goals. And then finally, our overarching goal is to develop an asymptomatic viral infection likelihood assessment for targeted roguing, because currently the only tool that the growers have right now is that active sampling, which is really quite expensive. And by the time they see the symptoms, it's too late to stop the spread. So we really want to be able to capture disease in its asymptomatic phase. So now moving on to our second case study. So now we're going to fly over the country. So I'm going to fly over Indiana, wave to all you guys. That work is all being done in California. And now I'm going to talk to you about the work that we're doing here in New York. So this is our project to build a closed loop between autonomous ground robots and high resolution satellite imagery. And this work is funded by the Cornell Institute for Digital Agriculture. So I'd now like to introduce you to my friend that's been hanging out in my virtual background. This is grapevine downy mildew. This is an omycete pathogen, Plasmopara viticola. And to be honest, it's really the #1 limiting factor to production in the Eastern industry. And we have to spray so frequently for it. Now I come from potato late blight and I saw intensive sprays, but it is nothing compared to what we have to do to manage downy mildew here in the Finger Lakes. We make sprays on 7 to 10 day intervals, and this is because it has a really explosive growth potential. I saw downy mildew go from nothing to, oh my God, I think I need to pull out Ridomil to save my vineyard in only 7 days. So it is really a devastating pathogen. Now, as a great pathologist, I can't help but love it a little bit for that damage that it caused, but it is truly a devastating pathogen to our industry. Additionally, it really sometimes feels like I'm looking down the barrel of a gun or something, because we've only got 4 modes of action available for control. And we recently discovered resistance to one of our most important tools, which is our FRAC 40. So those are our Revis, as well as our Zampro. That was discovered in Virginia in 2016. Now, we started surveying this summer to determine whether or not we might have this resistance as well in New York. But the overall summary here is that for downy mildew monitoring, because of the nature of its disease, its life cycle with the explosive growth potential, and the fact that it's almost ubiquitous in New York, we need high spatial and temporal resolution for successful early intervention. Now, when we manage downy mildew, we say if you haven't started spraying by post-bloom, you just might as well stop. Because if you haven't sprayed by the time you see downy mildew, you're kind of gone. But we hypothesize that if we can find the very first one, then we could actually manage according to that finding. But to find it, we need a couple different things. So this is those are the thoughts that I want you to keep in mind before I get to what we've actually been working on. Now, coming into this job, there were a couple of existing powerful tools that I knew I was going to be able to leverage. The first was MyEV. So this was an open source tool for vineyard data visualization that was built by the Efficient Vineyard SCRI. Now, this is an awesome tool that our growers are familiar with working with. It takes proximal sensing data and automatically maps it and puts it into a really nice, easy to use format. So our growers are comfortable working with spatial information in this tool. It's open source. It's available to any researcher, any grower who wants to use it to visualize proximal data in a vineyard. We also have DMCast. So this is a grapevine downy mildew prediction model that's integrated into Cornell's newest system. This is a network for environmental and weather monitoring. So we knew that we have this tool for visualizing data that our growers are comfortable using, and we have this environmental prior that we could potentially integrate in any sort of detection model. So these are the tools we knew we had to leverage to solve this problem. And then a coronavirus pandemic happened. So I first want to introduce my colleague here, Yu Jiang, who's another assistant professor. We started within 4 weeks of each other, and we started talking. We have similar areas of expertise. I work with spectroscopy from the biological side, and he's worked with spectroscopy from the engineering side. So we started talking about what we could potentially do to work together. And then the shutdown happened, and we thought, wow, we have no friends. Do you want to talk to each other to stay sane? We have no lab mates yet. So we talked every day, and we just started brainstorming what we wanted to do, what we wanted to build our programs into. And we came up with this idea for a closed loop, or integrated proximal and remote sensing. So this is a partnership between our 2 programs that's been aided, supported, and supplemented by the Vitis Gen 2 SCRI. So on the ground, so the overarching goal of what we want to build are ground detection robots that are capable of really, really accurately identifying disease. But where do we send these robots? That's where the satellites come in. We want to use these high spatial and temporal monitoring tools, our existing resources through DMCast, where we know the environmental conditions that are necessary to trigger downy mildew infection, as well as this existing spatial visualization tool. So we want to be able to build a system that can identify the best areas to go scout on any given day. And we want to build autonomous robots that can do that scouting for us. So that's the overarching goal of the project. Now, the first step here, though, was to develop the deep learning model for the little robot. So the robot is named the Phytopatholobot, after phytopathologist. He's the first one of the Phytobot system. Yu's program will be building more and more Phytobots, but when they're looking for disease, we call them the Phytopatholobots. So the initial deep learning model that he built for me had about 82% accuracy. And this was still quite good for an engineer who labeled all these images, and he had never seen downy mildew before in his life. Now we were able to improve the model accuracy by about 13% by bringing in my field specialist, Dave Combs, who's been scouting for downy mildew for over 20 years. So just by having that expert annotation, we were able to increase our model accuracy by about 10%. So now our little Phytopatholobot deep learning model is capable of finding downy mildew both on clusters and the foliage with just about as accurate as Dave himself is when he looks at these images, which is pretty impressive. So this work is in preparation, so keep your eyes peeled for a bioarchive post hopefully soon. So what's next for the phytopatholobot? So next summer, he'll be getting a bunch of cool new toys on him. We're putting an imaging spectrometer onto him, as well as a thermal sensor, as well as a LiDAR scanner. So he's going to be able to map and reconstruct vineyards in near real time. So he's getting new toys. He's also going to have some co-robotics. So he'll be integrated with various environmental sensors that we're putting into the vineyard, as well as aerial platforms too. So we're building, I gotta be honest, the engineer is doing all this. I couldn't explain to you the deep learning model architecture even if I tried. So if you are interested in hearing more, I invite you to invite Yu Jiang. He's a really engaging speaker, and the work he's doing for plant pathology is really just outstanding. So phytopatholobot's gonna have cobots. And finally, he's going to have near real-time detection. So he'll be able to go into the vineyard, plug himself in, and then automatically develop a map of where downy mildew was. And what we'll do with this data is to assess the plant phenome, both the physical characteristics and the biochemical characteristics via the imaging spectrometer. So our vision for this, we started to do a bit of this summer, though we did run into some problems with COVID, but we did get a couple of really good data. So in the background here, this is our Cornell Pathology vineyard. These are my shards where I do my fungicide efficacy trials, and these are really our home base for all of our New York-based sensing work. So our vision for this is to send the plant patholobot out. He'll find disease and generate a map. And with that, we'll utilize this to make different architecture recommendations for what different commercial satellite providers can actively do right now in terms of disease detection, and advise them how to do this better moving forward. So that's our overarching vision and how we're building towards having this closed loop project. So some of the cool things on the horizon for this project is that we're working with some cool synthetic imagery. So this is imagery that was generated by Bruno Aragorn, who's a new postdoc at JPL. He's currently stuck in Saudi Arabia, though, because of the coronavirus pandemic, which is unfortunate, who built us this really cool synthetic imagery. So using his novel synthetic band approach, we're able to increase the spectral resolution while maintaining spatial resolution. So that's what's so powerful of our SkySat imagery, is that it's at 50 center resolution. But as I mentioned before, it has very low spectral resolution. So using his approach, we're able to merge it with Sentinel, which has a better spectral resolution, so we can get that really nice spatial resolution while getting higher spectral resolution as well. And we hypothesize that this can improve our detection accuracy, and we're building this into our closed loop project. Another little side project that came out of this is that throughout the receiving all these different scouting data out of California within our own vineyards, we were like, this is kind of hard. It's really hard to digitize all this information. So our technician, Josh Evans, who shared with the USDA, he is currently building us an open source tool for automatically georeferencing scouting data. And we're planning to release this tool as a QGIS plugin so that anyone can use it as well as through MyEV. So MyEV was initially built for proximal sensing data, so it has to be modified in order to take in remote sensing data. One of the challenges we had with MyEV, well, not challenges, it's a great tool, but one of the obstacles we had to overcome was that it relied on it inputting a list of points that you would gather from your proximal sensor. Now, what if you're starting with remote sensing data where you don't have that list? You somehow need to generate those points in order to get the vine-by-vine health values. So we're using this tool to help solve that problem, but also to help solve the applied problem that as plant pathologists are gathering more and more scouting data, we're working across scales. It's going to be really helpful for us to identify the individual plants, especially with our perennials where those individual plants are kind of big and easy to identify. So Josh's tool will hopefully be helpful for everyone else as well as our own interests. And these works sum together towards a pretty cool proposal that we submitted to NASA's Commercial SmallSat Data Analysis Program. This is establishing a partnership between Cornell University, particularly Cornell AgriTech, and NASA Harvest. So NASA Harvest is NASA's food security and agricultural applications arm. It's outsourced to the University of Maryland, and it's pretty new. They only established it in 2018. So we summarized our closed loop project and proposed it, and we're hoping to hear back. Good news soon. So now the overall intended outcomes from our work. We're building a DM satellite-driven scouting algorithm that could be used by all New York growers in order to identify the best areas to scout that will be integrated into MyEV and can be used with or without the scouting robots. So the advantage of the scouting robots is that they'll eventually be able to teach the satellites better, in that this is actually how much disease was in this pixel. as well as, and the satellites can teach the robots where to go. But some growers may not want to invest in the robots, though we do hope to offer them at a price that's under $10,000. So the Phytobot system will be offered by the Care Labs Phenotyping Service Center. They have a number of cool offerings. Yu's still working on his website. I promise to share it once he's got it up. But they're actually going to be offered for purchase by other academic labs, individuals who just happen to want a Phytobot, as well as growers. And then you can use our scouting algorithm with or without the system. We're also going to be able to make architecture recommendations to commercial satellite providers, both through the NASA grant that we're doing and also through relationships that we've built through doing this work, in order to make recommendations how they can offer disease detection services commercially, but also to build and put into their pipelines for next generation development systems that are ideal for disease detection. My EV will be adapted for remote sensing through a couple different cool tweaks that we'll be making to it. But finally, the overarching goal of this work is to help preserve our high efficacy but resistant prone chemistries for use only when and where most needed, especially for grapevine downy mildew, where we're already facing such a losing battle. So we hope that these technology-enabled systems can help us preserve the existence of our chemistries that are so effective at downy mildew control and extend their lifetime further. So now I do think I have a bit of time. So here's just 2 slides, I promise, about cool work being started by our postdocs. The first is the Plant Protection Sensing Project. This is being led by Dr. Nikita Gambier. And this project, she's asking the question, can we use in situ and imaging spectroscopy to monitor for fungicide activity? So we know that these sensors work because the light directly interacts with plant chemical bonds. We hypothesize that it can interact with these synthetic bonds as well. So she's going to be investigating whether or not she can quantify SAR activation, as well as quantifying synthetic fungicide residue, as well as activity and efficacy with in situ spectroscopy, with the goal of building up towards using the imaging spectrometer that will be placed on our autonomous robot. So Nikita is starting in 2 weeks, and I'm super excited for her to join us. And next, we have our cool project that was funded by NASA, which is really exciting. We received an interdisciplinary sciences grant to study soil-borne pathogen dispersal on global dust currents. So this cool project is super multidisciplinary. It involves comparative genomics, earth system modeling, and atmospheric dust transport modeling, as well as remote sensing and plant pathology. So I'm really excited for this work. We made an offer to an outstanding candidate, and hopefully she'll say yes, because we're really excited to see the next steps of this work. So I know it sounds kind of crazy, right? Soil-borne pathogens on global dust currents, but it turns out that the foundation is all there. So we know that agricultural dust actually does contribute to these dust currents that go across the world. These arrows here are the dust currents that actually go around. And we know that agricultural dust aerosolizes with these currents, and we know that when it aerosolizes, it brings with it the microbial constituents within the soil. And we have identified plant pathogens within atmospheric dust samples. So we're hoping that through this project, we can develop a disease surveillance system that when SBG goes online, we can plug right into it. So we'll have the foundation set for monitoring for soil-borne pathogen dispersal. And when we have SBG, we'll actually be able to actively monitor for active disease due to this dispersal. So now just a couple of summarizing thoughts about studying plant microbe interactions at scale. The first is that imaging spectroscopy is a really powerful tool for plant disease detection, as well as management intervention. It's a powerful tool, but you can really think of it as a very sharp knife. It's a very powerful tool, but it will cut you if you're not careful. So you do have to consider how you use it carefully. The first consideration is that plant disease sensing is a function of plant sensing. When you're using a LAMP assay to detect a pathogen, you're detecting the actual pathogen constituents. When you use spectroscopy, you are detecting the plant's reaction to the pathogen's influence on it. So this is why it's so important to consider those environmental and host dynamics when you're doing plant disease sensing. It's different from our active samplings. It's a function. Next, when you're choosing what sort of spectroscopic data or what sort of spectral system you want to deploy, and if your goal is to develop an effective tool for use, you really need to choose this tool with respect to underlying disease physiology as well as pathogen lifestyle. You want to choose a tool that is effective for the latent phase of your pathogen, the temporal, spectral, and spatial requirements that you need for effective monitoring if you want to develop something that's useful for early management intervention. So that's why you also want to consider your 0 or non-zero management threshold tolerance when you're choosing what spectral system to invest in, both investing financially and investing time and people hours into. And finally, as my summarizing thought, early disease detection with remote sensing has the capacity to improve all three aspects of sustainability: the financial, the environmental, and the societal aspects of sustainability in grape and other specialty crop systems. So now, if you'll forgive an assistant professor for ending on an advertisement, we are recruiting for our closed loop project. We're recruiting a PhD student with a computational background to start with us next summer or fall. Cornell is accepting graduate applications through December 1st, and we are accepting students both through plant pathology, our computational biology program, and through our partnered care lab through biological environmental engineering, as well as ECE. So that's the electrical and computer engineering. So if you're interested or know someone who's interested, please encourage them to get in touch with me. So now I think I do have some time for questions, but I'll also be hanging out in my Zoom room for the next 30 minutes after this talk if you want to stop by and ask the speaker a question. So thank you. Yeah.
Thank you very much, Katie, for the great presentation. So I don't think I have any question here from members of the audience, but I actually have a couple of those questions. Let's start with this one here. So what ground truth data are you currently using, and how are you making sure that there's an adequate agreement between the spectral information and the ground truth information that you're collecting?
That's a great question. So we collect a number of information. We collect incidence, we collect severity. It depends on the disease. So when we're working in California, we're just collecting incidence because it's more of a yes/no question when it comes to the viruses. When we're working with downy mildew, we're collecting incidence and severity over time. We use RTK GPS to ensure really accurate geospatial ground truthing. And for the most part, our proof of concept work is all conducted within our fungicide efficacy trials. So we have collected some data at commercial farms, but for the most part, we're still in the proof of concept phase. So we work in an environment, a vineyard environment. It's like a commercial vineyard, but it's one that we know very, very well and very intimately.
Fantastic. And so here we have a question from Cliff Whale. Do you know what compounds in these leaves you're detecting? So I think that has to do with how powerful the technologies can be, but so maybe we're not completely sure what physiological or biological processes are actually happening. So what do you think about it?
Yeah, so yes and no. So to an extent, so we do have foliar trait models that we can use that have been developed by terrestrial ecologists. I rely heavily on the Townsend Labs models. These are publicly available through their, I think, EcoSML. So this is where their models are available. If you work with in situ spectroscopy, you guys also at Purdue have John Couture, who's an outstanding chemical ecologist who could tell you much more about this than me. So we do know to an extent that we can detect phenolics, sugars, starches. We can detect lignin, cellulose, all these sorts of constituents. And then we can also hypothesize onto what sort of plant biochemistry is contributing to our diseased plant response through what's been developed over the years in terms of what different wavelengths actually interact with. So when I have a disease model that relies really, really heavily on 1660 nanometers, I know that that's likely linked to phenolics because 1660 has a direct link to that phenolic structure. There are other wavelengths that have links like that to nitrogen, lignin, all these other sorts of compounds. So we can both we can directly assess, we can hypothesize based on the spectral information, and then we can hypothesize based on the biological information. So if we're trying to sense a necrotroph and we have a model that's really, really reliant on the SWIR, maybe it's sensing the toxins that necrotroph is producing to kill the cells. So those are some of the things. So we do, to an extent, know what we are sensing, but to a lot we don't. There's so much that we don't know about plant disease sensing, which is why my program is, I say that we study the fundamental science because we're looking to push forward the boundary on what we know about plant disease sensing.
Thanks.
Very good. And
thanks for your question.
And I have here two related questions. Let's see. Okay, right here. So many diseases have similar phenotypes, welts,
Yep.
spots, but are caused by different pathogens. Can you distinguish among pathogens once symptoms are apparent? And so there's a related question. Can you make a difference between insect damage and pathogen damage?
Yes. So the answer is yes, but that we know that it's possible. We know at the basic science side that is possible. And we found this with in situ spectroscopy, both at the contact level and at the canopy level. But on the functional side, in terms of what can I say is possible right now in the field, we don't know that yet. We don't know what's scalable and what's being lost. So we know that it's absolutely fundamentally possible, but we don't yet know what we can or can't functionally do in a true vineyard setting. And that's a lot of what we do as well. So today my talk focused on our remote sensing work, so really like our scalable work, but we do quite a bit of ground sensing as well. And this is a lot of what Nikita will be diving into, as well as some of our affiliates that I mentioned on the Grape Spec Lab, such as Saeed, as well as Ugo. So they're really deep diving into the basic science on the in situ side about what we're actually able to differentiate, because that is a huge problem. These things look alike. But what we have found in the in situ side is that necrosis caused by a hemibiotropic pathogen and a necrotropic pathogen, we could differentiate with high spectral resolution sensors. So that gets back to that spectral resolution side. If you're using low spectral resolution, two diseased leaves are always going to look more similar to each other than they do to a healthy leaf. The more spectral resolution you have, the more likely you are to be able to differentiate them.
Very good. Another question here. If I planted out a huge F2 population, could I do genetic analysis of disease resistance from space?
From space? In theory, yes, you could. So the challenge with that is that you would not want to use planet labs. You want to get as high spectral resolution as possible. Right now, the only satellite offering D-sphere imagery from space is Prisma. This is from the Italian Space Agency, and it's offering postage stamps. And they are declining to take requests right now because they've been overwhelmed. So they collect random postage stamps, and you would just have to hope that your plot would be within it. Fundamentally, yes, this would be possible. In practice, you'd run into some problems. I would recommend using a different sensing approach, either an autonomous rover, potentially a drone, or an aircraft, actually. Aircraft are very reliable, but difficult to deploy.
Good. I think this question is related. So what is the potential for using hyperspectral remote sensing for identification of potentially resistant or less susceptible cultivars?
Anna, I've read your papers. I know that that's what your interest is. I think it has great potential, absolutely great potential. I think you run into the same problem that Damon asked, though, is that currently if you want to use like spaceborne systems. So I guess the two questions are fundamentally, would it be possible? Absolutely, yes. I think that that would absolutely be something you can do. Right now, in practice, we're just running into some problems getting the data that we need in the right locations. So yes, I think the potential is absolutely there. And there's been some really great work with different breeding programs too. There was an Catherine Hamsel-Meacham. She published two series of really great papers this year about developing proximal hyperspectral sensing systems for breeding programs. So I recommend checking out her work. She comes from Brookhaven National Labs. Thanks for your question, Anna.
Question from Speakers Club, Imperial College. Which crops are most challenging to survey with this technology? For example, could it be used to survey tree health in a forest to see if the natural environment is under stress?
So my answer is that my crops are the most challenging because they're the problem I have to deal with. But no, that's not true. I think that every sort of system has its own challenges and opportunities. So absolutely, yes, there is a lot of interest. Janine Cavender-Bars is doing some outstanding work surveying for oak wilt with the hyperspectral remote sensing. But it depends on the sort of tree forest system that you want to do. So tropical systems are really challenging to sense, but they're really interesting to sense as well. So my answer to your question is that all crops are, just like all dysfunctional families are all dysfunctional in their own way. All crops are challenging in their own way, but it's just about really understanding the biology of your crop and the biology of the pathosystems you want to sense. Nothing is impossible.
Good. I think this is an algorithm question. So let's see. Reactions of disease could be quite similar to those caused by abiotic factors. How can you teach your systems to distinguish both types of stresses? Lorenzo.
Yeah, see, that's the million-dollar question, Lorenzo. That's the million-dollar question. So this is actually where the deep learning models, I think, are really powerful in coming into play. So we have some outstanding machine learning models that I use quite extensively, our unsupervised anomaly detection, random forest for one. We're starting to try out isolated forests. Those are kind of fun. But I think actually the deep learning architecture, so our RCNN model, I think that it could be quite powerful in that it allows you to integrate different data at different steps of the decision-making process. So that's a good question. So that's what we're hoping to try to get into with the triaging algorithm that we're working to build, is basically to try to just basically do a quick categorization with a really powerful model. So that's what I'm working on, Lorenzo. I'll let you know when I find the answer. If you find it before me, please let me know because I'm really interested.
Good. Question from Mariela Fernandez. Have you explored the effect of different nutrient amounts in the plant and the detection of the disease?
Oh, that's a really interesting question. So I'll answer the question that your question inspires in me, which is that it's really, you can't use these trait indexes to 100% of the time tell you that something's diseased, because the original state of the plant is so important. Remember, with plant disease sensing, we're sensing a function of the plant. So we have to consider how these things change over time, but we might not always understand their starting state or their true healthy state. So we do that nutrient, we know that these nutrients change. And from what I've seen at this point, I've looked at thousands of disease spectra. For the love of God, I can't find a rhyme or reason when I look at total phenolics, which way it's going to go, but I can tell you that it is changed. Now, one of the interesting things, if we're going to talk true nutrients, we found that in our compromised leaf study about the upper canopy foliage, that our upper canopy leaves of the disease plants actually had higher nitrogen content, and they visually appeared healthier than the upper canopy leaves on our spectral systems than the upper canopy leaves of the non-disease plants. So that's kind of interesting. You'd think that these upper canopy leaves would actually be less healthy, but we found in fact that they, by our spectral measurements, they seem to be more healthy, yet they had lower canopy disease. Whether or not that was a response of the plant to try to outgrow the pathogen, or the disease preferentially was appearing in healthier hosts, maybe it was like, oh, this one's tasty. So we don't know the answer. So the dynamics of nutrient and plant disease sensing is not quite fully understood, but it's a very interesting thing to explore.
Okay,
Thanks for your question, Mariella.
One last question. Are you using oblique drone capture imagery? If yes, how are you stitching these images together?
I am not. We work mostly with our ground imagery that where we rely on our engineers to turn into useful disease maps for us, and we use satellite imagery as well. Someone did fly a drone across one of my vineyards today, and I think they use Agisoft, but I don't know. Sorry. Right. But thanks for your question.
I have another question here. So how long does it take to capture the process the images collected by the robot? And let's see if this process is done manually.
Oh, it takes forever. I mean, it takes the span of an academic research lab working during a global pandemic. So we're at the stage of development. We're still annotating the images to continually improve our deep learning models. We have all the slow pipelines because we're working on just building the system right now. That's the next phase of the project, and Yu Jiang has his student, Tai Liu, who's working on automating this so that it can be done in near real time. So right now we are not at near real time yet because we're still in the development phase, but that's what we're aiming to get to.
Very good. And I have a question from
Also, thank you all. These are wonderful questions. I really appreciate it. This is great.
I have two questions from so Dayong, maybe it would be best if you ask the questions directly because these are a bit long. So would you like to unmute your microphone?
Yeah, hi, can I be heard?
Hi, yeah. I can hear you.
Hi, thank you for the informative talk. So I do have actually two questions. So the first one is, quantitatively speaking, how long is the long latent period that you mentioned earlier in the talk, which seemed to be advantageous for pre-symptomatic detection of disease?
Oh, that's a great question. So with the viruses that we're working with, they can have a latent period as long as about 12 months. And that's in contrast. So that's not saying that that's what you need to do that. It's just saying that that was ideal for that, because we can only get average flights, you know, at best once a season, if that. They're quite expensive. Now, in contrast to downy mildew, where the latent period on the short side, when there's conducive environmental conditions, can be 4 days. But there's definitely some middle ground within there. So Pablo Zarca-Tejada's work with the pre-symptomatic figure that I showed from Nature Plants, that was working with Xylella fastidiosa in olive in Spain and Italy. And I think that one can have over a 12-month latent period as well. So I think that these aircraft Cadillac sensor systems are quite good for where you absolutely need detection. So maybe a pathogen like citrus greening or other tree diseases, long latent period diseases.
And just another follow-up question to that first question. So is there a specific time of the day which you prefer to collect those data points, which would more or less correlate with the physiology or the interaction between the pathogen and the plant? Or is it just about collecting frequent data along a span of time?
That's a really good question. So we target for solar noon because that's the ideal time of day for remote sensing. But there are diurnal dynamics in pathogens. So it's not quite as important as when you're trying to sense something like water stress, which has incredible diurnal dynamics. But we aim for solar noon, but that's something that, depending on the pathosystem, could be quite important.
Right. And my final question would be, based on the aerial images you shared with us, those images over California, I've noticed that you used MSAVI as your vegetation index. So was this confirmed with other types of vegetation index that led you to use the MSAVI to have the greatest agreement with your ground truth?
Oh, no. So we used MSAVI too, just because that picture that I showed, that was just kind of like a here's what it looks like with a vegetative index. So MSAVI 2 we prefer to use in California because the soil is so reflective. So MSAVI 2 is basically NDVI, but it's calibrated such that it can handle high reflective soils. So that's why I prefer to use that rather than NDVI when I'm showing images from California. So we did not do any sort of compare and contrast in terms, because we were mostly using that as like, here's an example of what it looks like from a satellite. We're mostly interested in our hyperspectral data.
Right. Thank you so much.
Yeah, thank you for your questions.
Thanks.
Very good. So since we are recording this session, I was also asked where the file is going to be uploaded. So I don't think we have an official site, but I'm more than happy to upload it on my website. And so if you're okay with that, we'll process these and then we'll try to upload it as soon as possible. So I'm just sharing the link of my website so you guys can check it out pretty soon, and then it should be uploaded in there.
Awesome.
So
thank you so much for the invitation to speak. You guys have been a great audience, and these questions were super wonderful. I'm going to open my Zoom room till 5 o'clock when I have to go to my students. He's getting an award. Very proud of him. Proud PI. So I'll be there for the next 22 minutes if anyone wants to ask the speaker a question. Thank you for the claps, Felipe. I see you.
Well, thank you very much, and thank you for joining us today. It was a great presentation.
Thank you all. Thank you so much.